we're going to look at all the events that lead up to muscle contraction followed by muscle relaxation. To do that, we need to go to the neuromuscular junction, or the synapse between a motor neuron and a muscle fiber. So the motor neuron is bringing impulses to the synapse and the muscle fiber is receiving it. So we'll start with an action potential that arrives in the motor neuron and it arrives at the synaptic knob. This is sensed by voltage-gated calcium channels in the synaptic knob, which open and calcium rushes in. Calcium causes exocytosis of the synaptic vesicles, which release a neurotransmitter called acetylcholine, ACH for short. Acetylcholine diffuses across the synaptic cleft and binds to its receptors on the muscle fiber. These receptors are called ACH receptors and they are ligand-gated sodium channels. So when these channels open, sodium rushes in, bringing positive charges into the muscle fiber, raising the voltage and thereby causing a depolarization. The muscle fiber is now activated. To be exact, it is the sarcolemma, which is the membrane of the muscle fiber, that is activated. So a local potential forms in the region of the sarcolemma, and we call this local potential an end plate potential. So we have just created an end plate potential in the sarcolemma of the muscle fiber. We know that local potentials cannot travel, so we need action potentials to be created. So here is a close-up of the muscle fiber. Shown in blue is the sarcoplasmic reticulum. The area between the orange lines is the T-tubule, where the action potentials will travel. So when the local potential, which is the end plate potential, reaches threshold voltage, it causes the opening of voltage-gated sodium channels all along the sarcolemma and the T-tubule. Those are shown in pink. So when these channels open, sodium rushes in, bringing positive charges into the muscle fiber, thereby depolarizing the sarcolemma and the T-tubule. This causes action potentials that are shown in the yellow arrows to travel all down the sarcolemma and all along the T-tubule. When the action potential travels down, it triggers the opening of voltage-gated calcium channels that are shown in green all along the sarcoplasmic reticulum. Remember, the sarcoplasmic reticulum is filled with calcium. That's its job, to store calcium. So when the action potentials come down, they induce the opening of voltage-gated calcium channels and calcium is released from the sarcoplasmic reticulum. There is more calcium inside the sarcoplasmic reticulum, so the calcium readily diffuses down its gradient from inside the sarcoplasmic reticulum to the outside. So in this way, calcium floods the entire muscle fiber. Calcium plays a very crucial role in muscle contraction. So before we look at the role of calcium, we should take a look at the actin filament, which is the thin filament in the sarcomere. Here the actin is shown by the blue circles. They represent the actin filament. The blue sash is the tropomyosin. You want to imagine the tropomyosin to be like a ribbon that covers the actin, a sort of a transparent ribbon that covers the actin. The troponin is like a button that's on top of the tropomyosin. If you were to press this button, you can shift the tropomyosin from its default position. Only calcium can press that button. So this is where the role of calcium comes in. So when calcium presses the button, which is troponin, it shifts the tropomyosin, which is what we see here. The tropomyosin has been shifted from its default position, and now it completely exposes the active sites on the actin. Now, these active sites are important because this is how the myosin makes contact with the actin. So here is a schematic representation of a sarcomere. The sarcomere is the unit of muscle contraction. Each sarcomere has tens of thousands of thin filaments and thick filaments. The thin filament is mostly made up of actin, and the thick filament is mostly made up of myosin. The thin filament has active sites. So the active sites are on the actin molecules. These are important because this is how the myosin makes contact with the actin. As we see these structures in pink, those are the myosin heads making contact with the active sites on actin. So the myosin head acts as a hook and the active site acts as the eye. So like a hook and eye, the connection is very firm. And when many hooks attach to many eyes, they can help to pull the actin filaments towards each other until they even slide past each other. This causes the sarcomere length to reduce drastically, and this is what contraction is. So when every myosin head makes contact with its active site on the actin, it uses 1 ATP. It needs this energy because every myosin head must actually lift before it makes contact with the active site. So when tens of thousands of myosin heads execute this power stroke in unison, contraction of the muscle is accomplished. Now the muscle needs to relax. For the muscle to relax, the muscle fiber cannot be excited. So no new action potentials arrive at the synaptic knob. So voltage-gated calcium channels do not open, calcium does not enter the synaptic knob, which means exocytosis of the synaptic vesicles does not occur. No more acetylcholine arrives at the synaptic gap. If this continues, then the levels of ACH will decline. Any remaining acetylcholine will be destroyed by a very important enzyme called acetylcholine esterase. This is ACHase for short. When acetylcholine esterase destroys any remaining ACH, there is no ACH to bind to its receptor on the muscle fiber. So these channels close. No sodium enters the muscle fiber, and so no local potential is formed in the muscle fiber. It is not excited. If the muscle fiber is not excited, it cannot generate action potentials. If there are no action potentials, then calcium returns to the sarcoplasmic reticulum. Not only does calcium not leave the sarcoplasmic reticulum, any calcium that's available also goes back to the sarcoplasmic reticulum. Because there is no calcium available, troponin cannot be activated. When troponin is not activated, then tropomyosin will shift back to its default position. So that's what happens next. Tropomyosin covers the active sites on the actin. Once it covers the active sites on the actin, the myosin heads cannot make contact with the actin, and so the myosin cross linkages are broken. Once these are broken, the actin filaments go back to their original position. They recoil and the muscle relaxes.